So that's what we had in store for our welcome message this morning. We have just a few minutes here before our next plenary session. So we can just take a few minutes to get started. So maybe I'll just introduce our session and ask our session speakers. I believe that Christine has made you co-hosts. So when it's your time, you can share your screen directly with us. Just as a reminder for all of our speakers, we'll make an announcement when you have 2 minutes left on your talk. So for our keynote speakers, that'll be the first warning at 18 minutes. Then we'll have another warning at 20 minutes to allow for 5 minutes of Q&A, and then a final warning at the end of the 25 minutes. And then for our other speakers, we'll have a warning at 11 minutes to give you sort of a 2-minute warning to allow us to have 2 minutes for Q&A at the end. So let's just take a few minutes to get set up. And then, yeah, we'll use those last 2 minutes if anyone needs to stretch their legs before we get started in our next plenary session at 9 a.m. Pacific time. So I'll stop sharing my screen, and Ben Bond-Lamberty, if you're already here, you can upload your talk.
Sarah, do you see my screen?
Yes, we do. Yeah, I can hear you. Okay. Great. So we'll just get started. We'll just give folks a minute to arrive, and then we'll jump in. See that you're not in your car today, Ben.
No, I'm actually in a room in a chair. So.
Okay. We'll go ahead and get started on. Well, yeah, we'll give ourselves a little time, start on time. So welcome to our third plenary session. So this one is on open science. So an opportunity to bring together and think about some exciting work that's been going on in the AmeriFlux community and more broadly. So we're going to have some talks on soil respiration, remote sensing, and then a global methane network. So our first speaker for today is Ben Bond-Lamberty. He's a research scientist at the Joint Global Change Research Institute, a collaboration between the DOE Pacific Northwest National Lab and the University of Maryland. His research concerns carbon and nutrient cycling with a focus on high latitude northern ecosystems. So with that, Ben, I'll let you take it away.
All right. Thank you, Sarah. And thank you hugely to the AmeriFlux for the invitation to kick things off today. It's really an honor. And yes, the talk here is integrating large-scale soil respiration and other data sets for understanding global change. And so there's a bunch of themes, a bunch of topics kind of wrapped into that title that I'll sort of be touching on as I go through here. So this is AmeriFlux. Probably most people know what I'm kicking off with, this idea of soil respiration. But if you've just come in off the street and stumbled on this talk, welcome. And so this idea of soil respiration it's a very poorly named term, right? Because it refers to usually the total flux of carbon dioxide between the soil and atmosphere. And it really, it started early 20th century was the first time someone had the bright idea to sort of stick a cup on the surface of the soil and see what happened. But really in the 1960s is when it really began to take off. And researchers, again, were starting to think about things like carbon budgets, say, and you could use a static chamber and measure how quickly an alkali absorbs CO2, basically. And then, 25 years later, the advent of infrared gas analyzers then gave us quick and easy ways to do this and to do these measurements. And it turned out people started doing them quite a bit, really. It's become a key measurement in many ecosystem ecology and carbon cycle studies. It's relatively, while not cheap, it's relatively straightforward to do and cheaper than many other things. The figure sort of shows the publications from Web of Science just on this broad topic. We've also recognized, again, with starting with sort of Bill Schlesinger and similar kind of folks in the 1970s, it's a very large carbon flux in the Earth's system, probably second only to photosynthesis in terms of its absolute magnitude. And it has lots of uncertainty. And I'm not putting up here sort of one of the classic, like, Friedlingstein kind of Earth system model 21st century figures, right, where the models in the second half of the century start to diverge wildly. But you can imagine it, as I've just waved my hands diagramming it. And a lot of that spread is driven by the terrestrial processes, in particular the soil response to climate change. So there's a lot of uncertainty here that we'd like to be able to understand. And up until, I guess, sort of 10 years ago, this was all what's called sort of the long tail of dark data. And in other words, most of all these estimates of say soil respiration were published in figures and tables in individual publications. And this idea of this dark data, in the sense that it wasn't open, right? It was closed, and you had to subscribe to the journal to even see it. And then it wasn't available. You had to laboriously sort of, if you're interested in something, you had a lot of research and typing ahead of you. And it's kind of interesting about this idea of this long tail of data. The figure is actually from this paper from a library sciences paper that you can sort of think about this as a proxy. This is NSF awards and the distribution of them. And there's this long tail that goes out, right, of relatively small awards. That's a very large fraction, actually. And as the author of this piece notes, any individual data set here coming out of one of these studies, may not be of wide interest, right? There may only be a few scientists worldwide who want to see a boutique data set, or an individual study. But there are thousands of these in that long tail, and at least this author, said it seems likely that transformative science may come from the tail rather than the head, rather than the $10 million things. We may be the fact that we can't access easily the data in the tail, in other words, is a significant problem. And this was the problem actually confronted as well by AmeriFlux and FLUXNET, right? That this is from Kim Novik's paper a couple years ago, and noting again the long tail of science. This is talking about eddy covariance, and this is sort of funding and degree of centralization, right? They've mapped out, but it's the same issue that before those kinds of networks like AmeriFlux, you had those data sets kind of dark and long tailish, and hard to do slowing, and making it harder to do good science, especially synthesis sorts of science. So back to the solar respiration side, I guess, people had done a few tables in individual papers, but my colleague Allison Thompson and I, 10 years ago, sort of made the first attempt to synthesize, to actually go through the tail and synthesize everything. And that's not everything. You have to make some choices about what you're going to do, or else you'll never finish. And that's a point I want to return to. But to synthesize data into an open community database. And so that's visualized sort of here. The day, it's sort of fair amount of data over 50 years, and that's been that was open from the get-go. And SRDBs, it's the sort of unimaginably named soil respiration database, and has been sort of periodically updated. And in fact, my colleague Jingfeng Xiao has just published, in Earth System Science Data, a new sort of restructured and greatly updated database. So that's really kept going, and it's cool. It was cool. It has been really cool, in fact, because I think it's fair to say it sort of kicked off a wave of studies, making these, thousands of observations available to the community. You had sort of model benchmarking, a lot of new efforts to estimate regional to global scale Earth system fluxes of, say, total solar respiration or heterotrophic respiration. So the figure on the right there is from like Jinsh's ongoing analysis right now, but kind of looking at people who've tried to models and data syntheses that have tried to estimate the total annual heterotrophic flux. Is the y-axis there in that's actually not right, in petagrams of carbon. And so you can see some convergence and some interesting dispersion. And the lower left figure kind of shows, that people have cited that original database paper, and those papers in turn have been cited. So it's sort of, I think, again, I think it's fair to say it really has set off kind of a wave of work, which has been really, really neat. And the last bullet there, it's made possible, again, this idea of not just at an ecosystem scale, but at larger scales, using some of these, for instance, so the atmosphere fluxes as a cross-check and a constraint on other parts of the carbon cycle, right? This, at the end of the day, we think we understand at some level, maybe what's happening in terms of the biogeochemistry and carbon cycling, and we think we should be able to make them match, and it's an interesting problem if we can't. And finally, and just I guess and this has only increased with particularly in the last, say, 5 years, with the growth of machine learning algorithms and people starting to apply them in imaginative new ways, artificial neural networks. This paper is from Dan Warner, Rodrigo Vargas, and colleagues last year, sort of used what's called quantile random forest, an updated version of the SRDB, to produce this global 1 kilometer map with errors. That's, I think, probably the new gold standard in a sense for a lot of like benchmarking and other work. So there's, so really neat work that's come out of it. There are problems though with this, okay, with it, for all that I think SRDB has been useful, both as a model for the open science in our communities and also in carbon cycle science. It has problems. It was built by a clueless amateur at the time. And so, there are decisions I made 10 years ago that I would love to get back if I could, and it's laborious, and it's post-publication, right? So it only uses the headline data reported in papers, sort of the data that generally that appear in papers, and that's a problem because there's a lot of data, we think, right, contained in the underlying data sets, the more detailed data sets, and that will inevitably vanish. We know this. Both, I think, anecdotally for most of us, right? Probably most people, once you've gotten, say, a decade into research, have had a hard drive crash or have had something bad and lost some data. And it's sort of scientifically too. So this figure is from a paper Vines et al. It's actually from the biomedical literature, but we have no reason to think that we're any different than them, right? And they tried to contact, they picked 500 papers at random and tried to contact the contact authors and request data. And, some fraction of those, they couldn't even contact the author. And if they could, was the author willing to share? And if the author was willing to share, did they have the data? And this sort of is the key, maybe the key figure, at least for me, that comes out of this, which says that, we lose about 17% per year of data extant, basically. Which, in other words, right, so if it's not deposited, say, in a permanent repository, if you're just dependent on the mechanism of reaching out to people to get their data, it's about a 17% per year loss of taxpayer funded, right, data. And, And for a model like SRDB, that's that intersects in some uncomfortable ways. In particular, I think, because the soil respiration side over the last sort of 10 years is going through kind of what eddy covariance went through 25 years ago, the advent of these automated systems that have begun to vastly increase just the amount of data. Right, so all these data coming out of these systems, measuring every half hour or hour, that obviously, no one's going to stick into a paper. You are the traditional model. You're dependent on supplementary information or writing to the author for the data. And the data at that temporal level of detail, there are some trade-offs, but at that temporal level of detail gives you opportunities to understand sort of fine temporal controls, sort of hot moments, control points, right? Things like that. Coupling with other carbon or sap fluxes, for instance, things like that, right? So all kinds of possibilities with that. And that's what really led then to a much more recent set of work with colleagues. And by a cool coincidence, it just went online this morning, actually, at Global Change Biology, which is this database. I pronounce it COSORE. Margaret Torn and I are going to have a discussion about exactly the pronunciation, but it's oriented towards continuous, so these continuous data sets, and tries to build on sort of, at least for me, some lessons learned. So emphasizes data provenance, traceability, has a lot more automated error checking built into it in QA/QC generally. It's set up for programmatic use from the get-go. So it comes as, you don't have to use the R package, but it comes as an R package. It's a real structured database. It has a much cooler name. And these are figures from this paper, again, that's online. And so we're really pleased about that. And all the data contributors, there are something like 85, 90 data sets, sort of an 8.1-ish million observations. So we hope it'll be a real resource, a sort of an open data resource for the community. Yeah, and just, so for example, right, Ruth Varner's data set, Harvard Forest data set from 2004 to 2007, growing season 8 chambers, kind of. So hopefully there'll be there's some opportunities to extend the science and look at just some questions that neither I nor maybe anyone else has thought of yet. So I guess then these two efforts, SRDB and COSORE, they're oriented around similar carbon fluxes, right? They provide sort of temporally divergent views, though, into that, and they have different structural approaches. Where they, and I should say the figure on the right is my colleague Stephanie Pennington's gorgeous visualization of the COSORE data over time by ecosystem type there. And these two, though, are the same. They have the same approach, a philosophy of open data, right? So, so they're both basically like CC BY4 would be basically the license. So in effect, it's please, here are the data. We request you cite, please engage. But finally, these data are publicly funded in a public trust. They also, and this is sort of my kickoff, I guess, for my last few minutes of this talk, they share similar philosophies that I think you can argue with the decision, but you need to be aware of the trade-off. And it's something like, don't let the perfect be the enemy of the good. They both take a pretty targeted approach, trying to be sort of as, in particular with COSORE, I think, as simple as possible, but no simpler in a way. And they make so on the one hand, we have the power of open science. Oh, and I wanted to highlight this example because it's super cool. This is high school intern this summer who is with us, Max. And he was interested in, we spent sort of a couple weeks at the very beginning of his 12-week summer, basically, 10 to 12 week, coming up with a good science question. He zeroed in on what factors control the balance of heterotrophic versus autotrophic soil fluxes. So, right, if we measure soil, the flux, soil respiration flux at the soil surface, we don't know a priori how much is coming from, say, plant roots versus microbes, and we have a bunch of ways to try to get at it. Some of them highly invasive or disturbance oriented. Some of them make a lot of assumptions, but we have a lot of data now collected on this, for in this case an SRDB, on the fraction of what percentage is coming from roots. Something like 700 observations people have reported. And so Max took this and then linked this up with other open source, open data sets out there. So you can see his slide up top. So a really interesting, really neat new data set on mycorrhizae associations globally, above and below ground biomass, test spawns, data set just from this year, things like nitrogen deposition, a bunch of other things. And, you know, for his final project, he produced this quite lovely map, which, you know, it's a summer project of a high school student. We'll need to investigate some things. I don't think that equatorial stripe is exactly true, but it's cool, right? Because these kinds of open data let someone with, you know, a motivated person like with Max's training to in a summer get at some really interesting questions. I think that, you know, lots of us could publish a paper on. And so we have then the databases he was using, right? We have these standalone databases, and I've sort of picked a half a dozen here examples. There's been this burst of activity of people making these great open databases targeting, you know, either targeted particular analyses or just sort of for the good of the community. And they have the potential, I think, to really accelerate science and extend what the things we can study. But they have limitations, right? They have, there's some downsides to this model. And this was really brought home to me recently, working with Christina Anderson-Teixeira to import SRDB, the soil respiration database, into her 4C database. And without going into gory detail, it was a crap load of work. And you know, it exposed some conflicting assumptions and inconsistencies. And there just are more general problems about can be hard to match up data sometimes if you have sort of lots of different data sets. And there's always the risk that small kind of bespoke databases won't be well supported, I guess, maybe by the community or by institutions.
2 minutes, Ben?
I have about 1 minute.
Yeah.
Okay, sounds good. But I guess I think it's worth pointing out that integration has costs too. And you know, you have more complexity, means more time, means more cost, means fewer data in the database for the same amount of effort. Or you know, if your requirements are high, that may impose a cost. You know, to go back to the COSORE example, that was spun up from the idea to submission in about 15 months. And that's, you know, you pay a price for how narrowly it's targeted, but there are some benefits, and that's important, right? To go back to slide 16, you know, kind of tick tock, 17% a year. And as Elizabeth Wolkowicz and colleagues, you know, made the nice point, you know, in an era of global change, our measurements are irreplaceable. They're not reproducible. And so that tick tock weighs heavily, I think, on all of us. But this balance is something to think about. I think this, you know, the long tail grassroots efforts versus sort of the potential of bigger, some more centralized efforts, and they're both necessary, I think. But it's interesting, I think, for the community to think about the balance, and in particular, how I think all these efforts, these open science, open data efforts, particularly benefit junior researchers and researchers and scientists with fewer resources. And that's a benefit, you know, it increases access for the entire community, and that's a good thing. Thanks to a whole bunch of folks, both students, collaborators, and agencies and efforts that have provided funding for this. So sort of some of the things I talked about or other efforts that contributed intellectually to this.
Wonderful, thank you, Ben. We have about 5 minutes for questions. Ankur says, open data evangelist Ben Bond-Lamberty, preach the world. Any comment or any questions for Ben?
Oh, we have someone with their hand up. I'm going to unmute.
Oh, yeah. Thank you.
Hi. So I was just wondering, since majority of the data points actually come from the northern latitudes, and there are very few data points that actually cover the tropics. So I was wondering how well can the data points from the northern latitudes capture the dynamics in the tropics?
It's a great, thanks for the question. It's a really good question. This is not, and you're exactly right, both for all the data I showed here, and this is common to other, all kinds of other geosciences databases as well. My guess would be not very well, right? And I think Dave Schimmel and colleagues a few years ago had a paper sort of pointing out, you know, the asymmetry between, and other people again have pointed this too, but the asymmetry between where our carbon cycle uncertainties are and where our measurements are located. And so, yeah, no, that's a real problem. Like just from a data constraint, you know, point of view, we do not need any more mid-latitude temperate forest observations.
Thank you.
Any other questions? I have a question for you, Ben. How do you encourage folks who might be concerned about sharing their data, you know, openly, especially maybe more junior scientists? What sort of advice do you have for folks that might be a little reluctant to share data openly?
So I think it's a good question. And I think it's too easy, I think, when you're sort of established and mid-career to blithely kind of dismiss it sometimes, right? And to sort of get up on your open data soapbox. I think, that said, I personally think, you know, we're human, and most scientists in my experience tend to think that everyone is watching what they do. And that does not tend to be true, actually. Like, the odds, in my experience, the odds sort of being scooped or having your data used are low. What certainly is possible, though, is that someone will grab a database, say like COSORE, and not, you know, I can't control if they properly cite, as we request, all the underlying publications. But I guess my answer there would sort of be, you know, if you don't contribute your data, you're not going to get cited. And if you do contribute your data, at least you might get cited. And the benefits, I think, and that's one thing we tried to do with this 80 author and the GCB paper, and TRI and other databases have been really good as well, is trying to draw people in and strongly encourage users to, you know, to reward data contributors. Right now, it's an imperfect world, and co-authorship is the strongest reward we have.
Great, thanks. Sorry, I missed a few questions down here. Just maybe one person asked, would you want to comment on the comment or on the statement, The Futility of Soil Carbon Sequestration by Bill Schlesinger?
I don't want to comment on it because I have not read it.
Okay, sounds good. And then we had a question about how about the tropics? Can we perform similar synthesis?
Again, interesting question. I think we're getting there. I mean, the situation is improving noticeably, and you know, but my guess would be no. Like, we can't constrain things to the degree we can elsewhere. Right? The area is so big, the carbon fluxes are so big, and the measurements are so few.
Great, thanks, Ben. So we're out of time. There's a few more comments in our chat. If folks can email Ben or go find him during the poster session. But we'll move on to our next talk. So our next speaker is Jingfeng Xiao, who's a research professor at the Earth Systems Research Center at the University of New Hampshire. And can you go ahead and share your screen directly?
Okay, can you see my screen?
Yes, yeah. And again, I'll give a warning when you have 2 minutes left in your talk. Thank you.
Okay, thank you. Hi, everyone. I really enjoyed Ben's talk. I think many of us benefit from and also contribute to open science. I'm really glad to have this opportunity to introduce a part of the CIF-related work that we have been doing over the last 3 years also. Most of the work was actually done by my postdoc, Xin Li, who is probably at the meeting here today. A variety of methods have been used to quantify GBP globally, such as satellite-derived vegetation indices, light insufficiency models, process-based models, and machine learning approaches. Despite all these approaches and all the previous studies, there are still a lot of uncertainty in global GBP estimates. Take process-based models as one example. As a global GBP estimate could range from 100 to 140 petagrams of carbon per year. This is a substantial spread in GBP estimate. All these approaches make use of all kinds of input data, such as climate, soil properties, land cover maps, and so on. All these input data have significant uncertainties at the global scale. And can we use an approach that does not rely on all those input data? This way we could avoid many sources of uncertainty and potentially reduce the uncertainty in global GBP estimates. As emerging solar-induced chlorophyll fluorescence, or SIF, is perhaps such an approach. SIF is essentially the glow of plants under sunlight. It is an energy flux re-emitted by plant chlorophyll molecules, perhaps a few nanoseconds after light absorption. So light energy absorbed by plants has 3 pathways: photochemistry, heat dissipation, and a small fraction re-emitted SIF. So top right figure shows the spectrum of SIF emission. Its wavelength ranges from roughly 600 to above 800 nanometer. Because SIF is an energy flux re-emitted by plants during the photosynthesis process, so it is more physiologically based than vegetation indices. The retrieval of SIF from satellite data is a little challenging, mainly because SIF only accounts for 1 to 2% of the outgoing solar radiation at the top of the canopy. It is therefore a very weak signal. The retrieval of SIF has been focusing on the so-called oxygen A and oxygen B absorption bands, where the SIF signal is relatively stronger. The analysis I'm going to talk about are mainly based on data from the orbiting carbon observatory tube. OCO2 was launched back in 2014, and it has been collecting about 5 years of data. Compared to previous SIF missions, OCO2 provides more frequent observations and with the finest spatial resolution, roughly 1 kilometer by 2 kilometer. The retrieval of SIF focuses on 757 and 771 nanometer. As map shows, all the overpasses across the globe for a single month, July 2015 here. It does provide more frequent observations compared to previous missions. But even if we lump all the overpasses together for a whole month, we can see there's still a lot of data gaps across the globe. Let's keep that in mind. To use the SIF to quantify GBP globally, we really need to understand the relationship between SIF and GBP at the ecosystem scale. A bunch of studies have used the OCO2 SIF data and FLUXNET data to look at the relationships, and most of those studies are based on a single site or a couple of sites. Back in 2018, we did a global scale study. We screened over 800 FLUXNET sites for the concurrent availability of OCO2 SIF data and FLUXNET observations. And eventually, we were lucky to obtain data for a total of 64 sites across the globe, encompassing a large range of biome types. And then we use all those data to look at the SIF GBP relationships. The figure here shows the relationships of OCO2 SIF with FLUXNET GBP for both midday and daily timescales, and for the 2 wavelengths. With the midday timescale on the top, the daily timescale at the bottom, and 757 nanometer on the left, and 771 nanometer on your right. For each time scale, the relationship was stronger for the 757 nanometer than for 771 nanometer. So we used the SIF at 757 nanometer for the following analysis. For each wavelength, the relationship was stronger at the daily time scale than at midday time scale. For comparison purposes, we also looked at the relationships between GBP and vegetation indices, such as NDVI, EVI, and NIRV. It turns out that OCO2 SIF at 757 nanometer was more strongly correlated to GBP than those vegetation indices derived from MODIS. This shows why the SIF technique is really promising. Then we look at the biome-specific relationships. SIF was strongly correlated with GBP for all biomes except evergreen broadleaf forest. For the tropical biome, we had a very limited number of data points. This is really encouraging. Given the strong SIF-GBP relationships at the ecosystem scale for a wide variety of biomes, we might be able to directly use a single satellite observation, SIF in this case, to estimate GBP. This way, we could avoid the use of many input data such as climate, soil properties, and so on, that we normally use for the estimation of GBP. This way, we could avoid many sources of uncertainty at the regional to global scale. More interestingly, it appears the slope of the relationship is pretty much the same for almost all biomes. And if we force the regression line to pass the orange, then there is no significant difference in the slope between any two biomes. So it appears there might be a universal relationship between the two variables at the ecosystem scales across a wide variety of biomes. This finding has really important implications. Well, it indicates if we use SIF to estimate GBP, there's no need to know the vegetation type of each grid cell or each site. So we normally use land cover maps to identify the vegetation types of grid cells. And land cover maps are typically based on satellite data via image classification. And image classification has a substantial uncertainty itself at the global scale. If we can use such a universal relationship to estimate GBP, we can also avoid the uncertainty associated with land cover classification or even a land use land cover change. So with these established SIF-GBP relationships and OCO2 SIF soundings, can we map GBP globally? We are not quite there yet. The OCO2 data still spatially sparse. Of course, we could aggregate the individual soundings to cause resolution grid cells, but even if we use like a 2-week window, there could still be a lot of data gaps across the globe. So to map GBP globally using SIF, we really need to have spatially and temporally continuous SIF estimates. Therefore, we used a machine learning approach to develop such a spatially and temporally continuous dataset. And we use EVI from MODIS and PAR, air temperature, and VBD from a meteorological reanalysis dataset to help predict OCO2 SIF. We used over 2 million OCO2 SIF soundings to train our approach, our model, and about 1 million data points to evaluate our model. As evaluation indicates our approach could estimate OCO2 SIF fairly well with an R-square of 0.8, and the error in the SIF estimation is fairly small for the majority of the grid cells across the globe. The maps here, well, we then applied this approach to estimate SIF for each 0.05 degree grid cell and for each 8-day time step from 2000 to 2018. The maps here shows the spatial patterns of SIF and the seasonal cycles for a particular year. As a resulting global OCO2-based SIF product is referred to as GO-SIF. You could interpret it as GO-SIF. It just sounds well and easier to memorize. And this kind of spatially and temporally continuous dataset could have many applications at the regional and global scales. For example, we used the GO-SIF to look at the annual variability of ecosystem productivity at the global scale. And more recently, we used our GO-SIF data along with soil moisture and GBP from SNAP to assess the responses of GBP to the 2018 drought in the US Southwest, dominated by drylands. We've established safe GPP relationships at the ecosystem scale and spatially and temporally continuous safe estimates. We are now able to map GPP globally. With a set of safe GPP relationships that we establish to convert safe signal to GPP for each 0.05 degree grid cell and for each 8-day time step from 2000 to 2018. And it could estimate GPP fairly well at a variety of time scales. The map on the top here shows the mean annual GPP derived from our sample estimates, and the map at the bottom is the standard deviation or the uncertainty. The variability or the uncertainty is the largest in the tropicals.
2 minutes. Thank you.
Okay, thank you. We also compared our global GPP estimates against those from the literature. It appears our estimate is somewhere between those from non-process-based approaches and those from process-based methods. Well, in summary, what the safe technique is really promising and encouraging. We might be able to use a single satellite observation, like SIF, to estimate GPP at regional and to global scales. And we might be able to avoid many sources of uncertainty and may lead to GPP estimates with reduced uncertainty and computation. The emerging SIF technique is really one of the milestones in remote sensing of the terrestrial carbon cycle. This figure is from our review paper published in Remote Sensing of the Environment last year. Is a nice review of the advances in remote sensing of the terrestrial carbon cycle over 50 years. It's a really comprehensive review. It could be fun to read. Including references is only 37 pages long. And our GoSIF and GoSIF GPP products are both available at our data repository, data.globalecology.unh.edu. You're also welcome to use our datasets in your research for answering your science questions or benchmarking your ecosystem and land surface models. Compared to 2018, we now have access to a lot more OCO2 data. It's really time to revisit the SIF GPP relationships at the ecosystem scale. Besides OCO2 data, SIF observations are also available for new instruments. For example, CHOPAMI has been providing 7-kilometer data since 2017. And we recently have access to SIF data from OCO3, an instrument on board the International Space Station. There will be a bunch of instruments in space in the near future. For example, FLUX will provide 300-meter resolution data, and GeoCub will provide SIF data as well from a geostationary orbit. And these exciting new SIF observations, particularly FLUX, will likely make it possible to operationally estimate GPP solely from SIF. Of course, besides all those exciting SIF measurements, there are many other exciting satellite data as well. For example, eco-stress, we have been recently mapping GPP using eco-stress data as well. I would like to thank the support of NASA National Science Foundation. I also like to thank AmeriFlux, FLUXNET PIs, and the OCO2, MODISM, and the SMAP science teams. Thank you.
Wonderful. Thank you, Jingfeng, for your talk. We have time for 1 or 2 questions. I see a question here from Kyle. So thank you for your talk. Some recent work has shown relatively linear and ecosystem type converging relationships between NIRV and GPP. How do you see the advantages or drawbacks of NIRV versus SIF for GPP upscaling?
Well, this is a pretty good question. NIRV is a relatively new index, and it is really popular now. We actually looked at that index in several of our studies. NIRV has a really good performance compared to NDVI, and it might be able to have a slightly higher performance than EVI. But based on our analysis, well, SIF, especially SIF data from OCO2, is slightly better than NIRV.
Great, thank you. And we'll take 1 more question. There's more questions in the chat, but you can I'll continue those conversations after your talk or in person in Gather as well. Any of the new satellites game changers for SIF measurements? If so, why?
The game changers, I think, well, the two instruments are the Chopommy and FLUX. FLUX will be launched in a couple of years. Especially FLUX will provide graded SIF observations at 300 meter resolution. That would be really cool. So it's not like OCO2. OCO2 provides individual discrete SIF soundings. With the data from FLUX, we will be able to map GBP spatially and temporally continuously. That's really a cool data set. And another game changer could be the instruments on board the International Space Station, like OCO3, and those instruments on board on the geostationary orbit. Those instruments have diurnal sampling capability. This way, we could use those data to look at how photosynthesis might change over the course of the diurnal cycle.
Wonderful, thank you. And thank you again for your talk. If you'd like, there's a few more questions for you that you can respond to in the chat or throughout the rest of the meeting. So thank you very much. We'll go on to our next talk from Xian Wang from the University of Arizona on advancing dryland carbon uptake using new satellite-based indices.
You guys see the slides?
Yes.
Okay, great. Let me. Okay, cool. Thank you. My name is Xian Wang, and I'm a PhD student in Bill Smith's lab, and I'm from University of Arizona. Today, I'm going to talk about near-infrared reflectance and chlorophyll fluorescence capture complementary aspects of dryland vegetation productivity. I think this is some answers to the questions in that chat. Dryland covers more than 40% of Earth's terrestrial surface. It regulates trend and variability of global gross primary production, which is GPP. So left figure shows that a semi-arid ecosystem contributes the most to the interannual variability of atmosphere CO2. And with climate change, drylands are also expanding, so it will have more power to impact the global carbon uptake. From a remote sensing perspective, there are key limitations when you use vegetation greenness index, such as NDVI, to estimate the dryland production. So the left figure also showing the high heterogeneity of dryland ecosystems with a relevant percentage of tree cover, grass cover, and soil background. So this high heterogeneity and the soil background actually can reduce the accuracy of NDVI or the vegetation greenness index. Also, evergreen or hardy deciduous forest is a typical vegetation types in drylands, which is difficult captured by the greenness index because most of them stay green during most of the time of the year. So these limitations will contribute to, potentially lead to only 31% of GBP interannual variability when you use satellite greenness-based GBP products. From plant physiological process, the greenness is an indicator of photosynthetic capacity, which is APAR here. So we want to accurately estimate the GPP, which is just the one pathway of this APAR. So the second pathway would be the non-photochemical quenching, which could be indicated by the photochemistry reflectivity index. The third pathway is fluorescence. It is released during the light reaction when the vegetation doing photosynthesis. And we further use the NRV instead of NDVI to indicate the APAR because NRV has less impact from the soil background and also the vegetation structure scattering. We exclude the PII from this research because the low accuracy of current available satellite PII data. We focus on this Chopommy safe data because I think it is the best safe data at current. The spatial resolution is 7 kilometer multiplied 3.5 kilometer, and the temporal resolution is daily. It's available from April 2018 to current. The other data set we include in this research are daily MODIS and our NDVI NRV with 500-meter resolution. The study area we are focused are 22 flux tower sites with GBP processed across semi-arid Western US. So we furtherly classified these 22 flux tower sites into tree dominant, grass dominant, shrub close, and shrub sparse. So our objective is to evaluate MODIS, NRV, and tropomi safe data capturing GBP across these 22 dryland sites compared to NDVI. We furtherly downscaled the safe data, even though we have the best safe data here, but I still have COSORE resolution when you compare to the tower footprint. We use the MODIS NRV to downscale the safe data so that we have 500 meters spatial resolution. And this method is adapted from the Turner's 2019 paper. We only focus the data in a 1.5 kilometer around the centered at each flux tower site. So this also works for NDVI and NRV. Here is our first result at the cross-site level. Y-axis is GBP, x-axis are for the different indices. So across these 22 sites, NRV actually best captures the GBP with R square equals 0.82. But when you classify it into different vegetation category, the raw tropomi safe data best correlated with GBP for the grass dominance, tree dominance, and close shrub dominance. And but they are with the different slopes. The reason could be the different escape issue of tropomi safe data from the different canopies. And then we don't have significant improvement when we downscale the tropomi safe based on the MODIS NRV data. So we are not going to discuss too many details about it. And all the safe data will be just raw tropomi safe data. And the last point here is that there's no indices captures really well about GBP at the sparse shrub size with low productivity. And for the seasonal dynamics, NRV and safe data are better than the NDVI, but with their own limitations. So in this figure, the y-axis is R squared between monthly GBP and monthly NDVI. So all these 22 sites are kind of scattering in this figure. But when you go to see NRV figure, you will see clear patterns here. So NRV actually is limited at evergreen sites. So at evergreen sites, you will see low R squares, but when you exclude these sites, you get mean R square around 0.2. And then the safe data actually is limited at the low productivity sites. So at these sites, usually the noise overcomes the tropomi real signal. So when you exclude these low productivity sites, you get mean R squares also around 0.7. Here we are showing time series of the different indices at two representative sites. So the first row is WKG. WKG is a grassland site with few percentage of mesquite trees. And the second row is SIM site, which is a savanna site with a mixture of grass and mesquite trees. So different color indicate different variables. Black is GBP, green is NDVI, blue is NRV, and red is safe data. So when you compare the data at these two sites, there is a significant decoupling at the SIM site between the GBP and the reflectance-based indices, which are NDVI and NRV. So when you transfer this time series to the scatter plots, so this decoupling thing will become a significant hysteresis loops when NRV or NDVI correlated with GPP in that yellow dots. So the yellow dots indicate the decreasing of growing season, and the green color is the increasing of growing season. So, but this is not really happened during between the relationship between the tropomi and GBP. So we think this is related to the relevant percentage of mesquite trees at SIM site. And this hysteresis loops not only happen at these two monsoon sites, but also happen at the two Mediterranean sites. Here we are showing the VAR and TOM. The two sites are in California. VAR is a grassland site with few percentage of oaks, and TOM is also a savanna site with with mixture of grass and oak trees. And here is our take-home messages. So satellite NRV and SAFE actually improved the predictions of GBP across the dryland sites. Especially the Tropomi SAFE shows improved predictions for the GBP dynamics across the dryland evergreen forest sites, but it also with their limitations at the low productivity sites. And then reflectance-based indices show significant hysteresis loops with GPP. The reason could be the relevant percentage of the trees at those sites. And then safe downscaled NRV is not significantly improved for detecting dryland GBP. The reason could be the safe contributes most noise at the low productivity sites. NRV contributes most noise to the evergreen or the hardy deciduous vegetation sites. Drylands, most of dryland sites belongs to these two categories, or even the mixture of these two categories. So we have been talking about the satellite level measurements to understand the vegetation dynamics. But for the satellite thing, we always use one instantaneous measurement to represent daily data. So actually, the vegetation have their own diurnal dynamics. So we have towers to measure high frequency safe data, NDVI and PII data. So here is a site we are showing the GBP has really significant diurnal signals through spring, summer, and fall. Especially during the summer, there's a midday suppression. So our tower safe data actually showing, actually tracks these characters really well. So I think this high frequency data could fill the gap for the satellite level. So I think this is all I have. And I also want to thank you to all these collaborators for sharing their data and also give me a lot of great feedbacks. Thank you. If you have any questions.
Wonderful. Thank you for your talk. So we have a few questions coming in, and feel free to continue to type them in the chat. We have a few minutes. So from Gabriella, we have, could you expand on the complications of downscaling the SIF product to EC towers? What about multiple EC sites located within the same 16-kilometer grid?
About downscaling? Okay, which question? Which question? Sorry.
Yeah, could you expand on the complications of downscaling the SIF product to EC towers? And what if you have multiple eddy covariance sites located within the same 16-kilometer grid?
Okay, so we actually downscaled the SIF based on NRV to 500 meters, right? But it does not show great in our drylands. For the SIF data, we have 16 kilometers, right? We actually exclude the pixels which don't have the same vegetation covers as flux tower sites. Is that I answer the questions?
I think so. Gabriella, you can follow up if you have any other questions. Okay, let's see. Yes, thank you.
No problem.
Any other questions?
Most there is no complication.
I can see a few parallel threads coming through. Any final questions in our last few seconds here?
Whose approach did you use downscale the satellite? Okay, I think the method I'm using is from Alex Tanner, who is from Berkeley, I think. Before now is a assistant professor in University of Washington, I think. There's a paper in 2019. Okay, so they were bit of citizen more based on
Oh, sorry.
Okay.
So Jan, were you answering a question? Sorry.
I mean, I can answer question when I can type to them.
Okay. All right. Sorry. I wasn't sure if it cut off on your end there.
No problem.
Okay. Great. Well, thank you very much for your talk, and we'll move on. There's one more question down here if you want to answer in the chat. In the meantime, we'll move on to our final speaker for the session. So we have, closing off our session, we have Kyle Delwiche, who's a postdoctoral research fellow at Stanford University. And she'll be presenting on FLUXNET methane, a global multi-ecosystem scale database.
Hi, everybody. Thank you, Sarah, for the introduction. I'm really excited to be here today. Thank you to AmeriFlux. My name is Kyle Delwiche. I'm going to be presenting FLUXNET methane, which is a new global multi-ecosystem data set. And I'm really excited to say that this data set is actually now available for download. You can download it now, and I'll go over that. But today I'm going to give you an introduction to what this data set is, and then an application of what we've done with the data. Okay, so first I want to start off with a bit of background, because this has been a project that's many years in the making. So this is truly a community-led product. It was initiated a few years ago now by the Global Carbon Project, with the ultimate goal of gathering enough EC data to come up with a data-driven, upscaled estimate of methane emissions to the atmosphere. We were funded by a grant from the Gordon and Betty Moore Foundation, and then we got some supplemental funding from the USGS PAL Center to not only to collect the EC data, but then to also start doing some other interesting science with the EC data once we had it. And so this project has been coordinated all along primarily by people in the Jackson lab at Stanford. And this has really been Sarah Knox's effort for a long time now. She has done a tremendous amount of work to reach out to PIs and contact them, to ask for methane data, to work with them to get that methane data processed. You know, we started with a model a few years ago where PIs would send their methane data directly to us, to Sarah, and then also Gavin McNichol at Stanford, and they would work with the PIs to get that data processed. We've now moved towards a model where the methane data are first hosted on regional networks, and then the PIs work with FLUXNET methane. But I just want to acknowledge that Sarah Knox and Gavin McNichol have both put in a huge amount of time on this data set. Along the way, we've worked with other groups. So we've worked a lot with the AmeriFlux, the management project. We work with EuroFlux, we work with ICOS. So all these groups have been really instrumental in getting this going. And in particular, I want to thank Yue and Danielle from AmeriFlux. They have worked with me to get FLUXNET methane live and available to the community. And they've both endured probably hundreds of emails from me in the past couple months. So thank you to them for all their hard work and making this happen. And then I also need to acknowledge that FLUXNET methane exists thanks to the data providers. So we have many people who have contributed to setting up the towers, to going out into the field, to collecting the data, maintaining the towers. You know, and this is PIs, this is postdocs, this is technicians, this is graduate students, this is undergrads. This is so many man hours have gone into collecting this data and to the willingness to contribute it to FLUXNET methane. So this data set exists thanks to them. And so I really want to acknowledge that. And you will be seeing more pictures throughout the presentation. We have a lot of fun photos. This is a list of the 100+ data providers that we're currently working with now to write up a paper that's almost ready to submit that describes the FLUXNET methane database. So again, thank you so much to all of these people. These people have really made this happen. All right, so what is FLUXNET methane? Currently, it is 81 sites with methane data. These span 22 countries all around the world, and this represents over 200 site years of data. And as I said earlier, really excited to announce that this is now live. So if you go to fluxnet.org and you click on the data tab at the top, the pull down menu, the last option there is FLUXNET Methane Community Product. So you click on that link, it will take you to the landing page and that will walk you through how to download the data and a lot of information about the data set. The data download process is very, very similar to the FLUXNET 2015 data download process. It's using the same infrastructure. So that's just went up a few days ago. So please feel free to go check it out, use it. Yeah, use it. It's really exciting that it's there. So what does this contain? So we provide half-hourly data and daily aggregations for all of our sites, except maybe 2 that have hourly data. Every site has standardized gap-filled methane data. And this was done by Sarah Knox. She has 2 papers, one in 2016, one in 2019, that cover the gap filling methodology for that. And then there's a lot of work that went into standardizing those data sets. We provide gap filled meteorological data. And I do want to point out that this is gap filled using community standards. So this did not go through the OneFlux pipeline since we started our effort a few years ago. So hopefully in the future, this will be wrapped into OneFlux. And so the Met data will be exactly the same as OneFlux Met data, but for now it is actually slightly different. So please just be aware of that. This is why it's a community-led project, we're calling it that. Let's see, so we have a lot of sites that have other data like water table depth, soil temperature. So we provide all of that in the FLUXNet methane data files. We have site classification. So Gavin and I went through every site and classified it either as, a fen or a bog or an upland or a rice site or, all of these different classifications. And we did that based on primary literature or site PI communication. And then lastly, so we have a metadata table that you will get when you download FLUXNet methane data. And this includes vegetation information. And so we have presence or absence noted of these 5 different vegetation types for all of our sites. And then we also have a PI chosen dominant vegetation type. And so hopefully that will be useful as you think about synthesis projects that you could do with FLUXNet methane. All right, so let's just take a little bit of a deeper dive into what this data set looks like. So we have, as I said, 81 sites. So this is encompasses, we have 2 lake sites. We have 7 rice sites. We have 22 sites that are we call upland, and we break this down more in the metadata, but this includes things like alpine meadows, it includes agricultural lands, it includes forests and different types of forests. We have 5 salt marsh sites. And then our largest category is freshwater wetlands, and we break this down into bog, fen, marsh, mangrove, swamp. So we have these 5 different types, 44 of them all around the world. The time span for FLUXNet methane is relatively young, not too surprisingly. Methane data measurements have not been around quite as long as CO2. So by and large, most of our data is from the past decade, a little bit before the past decade. And then we have more data in more recent years. You can see so FLUXNet methane currently ends at the end of 2018. And that's just because at some point we had to stop collecting the data and actually work on processing it and then getting it into FLUXNet methane so we can make it available to the community. And we do intend to hopefully in the future obviously expand that so it incorporates more recent site years of data. Now, if you look at the annual flux range for the methane, we see that there's a heavy tail on this. So the over 200 site years of data, the average methane flux across the entire database is about 17 grams of carbon per meter squared per year. The median that is about 10. So we have a lot of sites with relatively modest methane fluxes, but then we have a fair number of sites that are a lot higher, and even one that's over 100 grams of carbon per meter squared per year. So that was the kind of the overview of FLUXNet methane in general. And as I mentioned before, we have our biggest categories are these freshwater wetlands. We have 44 of them. And because we have so many freshwater wetland sites, and these freshwater wetlands are a really important methane source to the atmosphere on a global scale, we took the FLUXNet methane data and we did a subset analysis on these freshwater wetlands. And here's just some of our pictures of our lovely freshwater wetland sites from all over the world. The first question that we asked with the freshwater wetlands is how do our FLUXNET methane wetlands, how do they compare to global wetlands? We have, two pictures here that show some broad range of sites. We have one down in the tropics in Brazil, and then we have Hausen's site, which is currently frozen in this picture. So, there's a huge range of global freshwater wetlands, and how well are our 44 sites, how well do they capture this global diversity of sites is what we wanted to know. And so this work has been done by Etienne Fleury-Chenard in our postdoc in the Jackson Lab at Stanford. So thank you very much to him for all of this. So what he did is he looked at 4 critical drivers of methane flux. Now, obviously there are many more critical drivers. These are the 4 that we chose to focus on for this based on some preliminary work. These were important and had globally graded products. So he looked at enhanced vegetation index, he looked at air temperature, water index, and then latent heat flux. And so basically looked at these 4 drivers for every point around the globe with wetlands and compared, what the global distribution of wetlands looks like compared to the FLUXNET methane wetlands in terms of these 4 drivers. And he looked at the mean annual values for these drivers across the globe. And so that when you can do is you can look at this on the globe. So this map shows every colored pixel here is a pixel with, I believe it's greater than 5% wetland coverage. And the color bar represents what we're calling a dissimilarity score. And so you don't need to worry about the math of what that means. It will be in the paper that we're going to submit relatively soon. But basically, the lighter the color, the more representative FLUXNET methane is of that type of wetland. And then the darker the color, the less well represented that wetland is. So for example, you can see up in northern Canada, there's a large extent of wetlands, and by and large, those wetlands are reasonably well represented within FLUXNET methane. Same with Northern Europe and Russia. But then when you go down, you look at South America in particular, and then Africa, there are large expanses of wetlands in these areas that are less well represented in FLUXNET methane. And this is not surprising to us. The circles show tower locations in FLUXNET methane, and so we have very few in these tropical ecosystems. And we hope that this sort of dissimilarity analysis can show the way forward for areas and ecosystems that we really need flux towers and measurements from to try to improve our understanding of methane dynamics from these wetlands on a global scale. So thank you to Etienne for that work. And then the last bit of work I'm going to show you something that I've been working on. So this is looking at the seasonality of methane fluxes across our freshwater wetlands. And so this is just 2 of many, many examples of data from our wetlands. So this is the same time period for each wetland, and you can see the points represent the daily mean methane flux. The light blue are the gap fill data, it's the gap filling that Sarah did, and then the dark blue are from the actual tower data itself. And so you can see that we have really strong seasonal cycles, not surprisingly. So in the winter months, you get relatively low flux, not necessarily 0, but lower than the warmer months. And then in the spring, that flux starts to go up, some cases quite dramatically. You have a summer peak, and then the methane starts to tail off and go back down to the baseline. And you can see even just between these 2 plots, looks like the top plot, that spring onset of the elevated emissions looks like it comes later in the season than the bottom plot. And so what I did is I took these curves and I used an online software called TimeSet to extract seasonality parameters. And you can get many parameters. For example, you can get the start of the season, and by season I mean the elevated emissions season. You can get end of season, you can get peak values, peak timings, you can get a lot of really interesting variables related to the seasonality. And then because we have a global database, we can start to look at, okay, what are the global trends in seasonality and methane? Can we say anything on a global scale? And so I can do this for methane, but I can also do the same seasonality analysis for GPP, and I can do it for temperature, very similar process. And so I'll show you one of the figures from that work. And okay. Sorry, Sarah?
2 minutes, Kyle.
Okay, thank you. Okay, so what I'm going to show you here, so this is, so I showed you in the previous plot that the mark of the start of the season, so you can get the day of year when, for example, the methane season begins, when the methane flux starts to go up. You can do the same thing for soil temperature. So on this plot here, I'm going to show you the x-axis is the day of year that the soil temperature starts to increase. The y-axis is the day of year that the methane fluxes start to increase. And so anything above that dotted line means that your methane fluxes are lagging your soil temperature, and anything below that means your methane fluxes are starting to increase before soil temperature warms up. So if you plot the points from all of our site years of data at all of our freshwater wetlands, this is what you get. And you can see that almost exclusively methane does not start to increase at a site until the soil temperature has already begun to warm up. And the lag there on average is about a month. And then you can do the same thing for GPP. See, the picture here is a little bit different. So here it's, it's roughly synchronous, but with a large spread. So at roughly half of the sites, we have our methane flux go up before GPP starts to increase, and at roughly half the sites, it's the opposite, where GPP goes up first. So this is just an example of some of the, the really interesting questions and trends that you can start to look at with this FLUXNET methane data set. And we really hope that this data set will be valuable for the community to look at these sort of broad scale synthesis studies. And so I'd just like to wrap up, and again, thank you so much to the data providers. They are really the ones that have made this happen, and we're so grateful for their willingness to share their data, to work with us, to give us metadata, and to answer all of our emails. So thank you to them, and if you have any questions, I'll take them now, and you can also email me. My email is there. Thank you.
Wonderful. Thank you for that talk. We have a question from Adrian Rusha. Can you provide a citation for the dissimilarity methane figure? Or maybe just
yes, so the dissimilarity methane figure is we have a paper that we're submitting any day now, as soon as I have time, so either late this week or next week. So these figures will be in that paper, and then the paper also has methodology references. So you can, and if you need more specifics about the dissimilarity work, you can email me and I can put you in touch with Etienne.
Great, thanks. And a quick question from Josh. How far are we from a good global methane wetland mapping?
That's not really my wheelhouse. I'm not working on wetland mapping, and we do have people in our group who are working on that. So making progress, but yeah, I would say we're making progress.
We can ask Etienne.
Yeah,
the one
so Etienne is also working on that, so we can also ask him.
All right, wonderful. Great. Well, I'd like to thank all of our speakers this morning for their great talks. I learned a lot, and it was great to hear from you all. So that brings us to the end of our session this morning. We have a little break until 10:20 a.m. Pacific time.